Good morning, everybody. Good afternoon for some of you. It's my pleasure to be here and tell you the short story of the Microbiota Vault initiative. So I did my PhD in the UK and I returned to Venezuela, my country of birth, in 1990. And in 1992, I started working with indigenous communities in the Amazon. And that work extended up to 3 years ago, and also expanded to other places of South America, including Brazil, Peru, and Bolivia. And we have, throughout all these years, make a very solid teams of local scientists and doctors, workers. I'm sure people in Africa know the difficulties and challenges of working in rural and remote communities. So during my work studying urbanization, what is the difference in lifestyle and what are the differences in the microbiota that those studies started in after the 2000. We collaborated, and this is the first paper was with Jeff Gordon, comparing the microbiota of Venezuelan indigenous people with Malawi and the US. And we observed the strong association between urbanization and loss of gut microbiota diversity. This observation was repeated in other studies and by other people, other teams as well. At the same time, urbanization is associated with the increase of the modern diseases we have discussed yesterday, immune and metabolic. So we know that there is a relevance of the human microbiota for human health, because when you perturb the balance of the microbiota, the services or the functions of the microbiota are also perturbed. And then there is an effect on physiology. The same happens with microbiome of animals, microbiomes of plants, and at the level of ecological interactions in the environment as well. So we know that because the microbiota is disappearing, the collections are very valuable. These high diversity specimens that are in remote places, difficult to access, challenging because of security. I mean, South America, in addition to insecurity, has guerrilla. It's extremely challenging. Sometimes it's challenging to get there because you need an airplane or a helicopter. These specimens are very difficult to obtain. And if we reason that if industrialization is degrading the diversity of the microbiotas, and that is associated to increase in modern diseases, can we restore? Can we reverse this trend? And this will require those collections. Those collections are typically not safe. I mean, in my lab, my collection has survived a revolution that has been very impactful against the infrastructure of the country, very destructive. Then hurricanes, as you will see. And in general, even in any lab, jumping off a breaker or somebody disconnecting your freezer can really jeopardize the whole effort. So this is the story of the vulnerability of the collections in my lab as an example. So in '98, the revolution in the country started. In 2003, I moved to Puerto Rico for 9 years, where we went through 9 hurricane seasons. In 2012, I moved to New York, where I thought I had left behind the hurricanes, but Hurricane Sandy hit very strongly New York. It has been the biggest of the threats to my collection. It took us to recover the collections from a lab in a 9th floor in the dark during a week, moving down the specimens by stairs because there was no electricity. And it closed our lab for 11 months. We couldn't go back to our labs for 11 months. It was extremely disruptive. During this time, we were publishing, analyzing data, and publishing our studies on the microbiome and urbanization. In 2016, I learned about the Seed Vault. And the Seed Vault, it's an initiative that inspired the Microbiota Vault. It was moved forward. The father of the effort is Cary Fowler. He was able to assemble a team to make possible the materialization of a global seed vault, which is currently in Norway in Svalbard archipelago, because agriculture is doing to plant diversity what agriculture and medicine and modern practices are doing to microbes, which is destroying the natural diversity. I highly recommend that you see this film, The Seeds of Time, which tells the beautiful story of the seed collection, the Global Seed Vault. So in 2017, I met with them. I met with the people of the Crop Trust, which is the nonprofit behind the Seed Vault. And then soon after, in early 2018, I met with Manuel Frankhauser from the C-Ray Foundation in Switzerland. And that since then, Manuel has been together with me, making the team grow, and his foundation and his effort has been really valuable on making the Microbiota Vault possible. We assemble a pioneer team. With the support of his foundation, we launched the first web page. We kept assembling collaborations during 2018. We published that Perspective in Science, which I just discussed. And then we visited together with Marty Blazer, we visited in the Swiss Alps some bunkers, which are candidates to be a storage place. We had the opportunity in 2019 to meet Cary Fowler, the Peru ambassador to the UN, because it also will require political support. We registered the nonprofit in mid-2019, and then we started efforts toward a feasibility study, which was carried out by Dominic Steiger since the mid-2019. We had meetings in Lisbon. That was the first meeting with the team, then in Berlin. Meanwhile, we also met in Oslo with Rob Knight, Dominic Kogand, and Tore Midtvedt. Tore has been moving efforts in Norway to also explore having a site there. We started thinking in the GloMiNe. We spoke with the Indonesian team in 2020 and with the Peru team. In mid-2020, the feasibility study was finished, and we started a bridge phase, a pilot phase, asking for support to foundations. We received support from the Gerber Roof Foundation, and later from the C-Ray, more support, Rockefeller Foundation. That has made possible launching the pilot phase. And we had our first microbiome, a global microbiome network symposium, the first GloMiNe with Peru in 2021, and this is the second one in 2022. The launch phase of the effort is including gut, human gut, and also microbes from fermented foods. But the idea is to really scale up to other sites of the human body, as well as microbiomes of plants, animals, and environment. So here we have the Microbiota Vault leading team. In this square is the pioneer team. We have people from very well-known scientists that you will recognize from the US, from Norway, Switzerland, Hong Kong, and UK. We have 2 Nobel Prizes in the team. One is Rich Roberts, who won the Nobel Prize in Medicine for his work on restriction modification enzymes. And the other is Jim Heckman, who is a Nobel Prize in Economics. And what is an economist doing here? Well, he studies the cost for countries and health systems that chronic diseases, non-communicable diseases, will represent for the world, and the incapability of the current health systems to deal with those diseases. More recently, this year, we incorporated Deborah Delgado Pugley, who will be a speaker this afternoon, who is one of the organizers of the first GloMiNe in Peru, Alex Kwarteng from Ghana, who is one of the organizers of this GloMiNe, Thomas Bosch from Germany, and Bob Goodman from the US. And Dominic Steiger is an officer of Evaluate Science, and is also an officer of the Microbiota Vault. He's also here. The launching team in Switzerland, you will also recognize them. Adrian Egli will speak today. Pascal spoke yesterday. Pascal Bonash, and Nick Bokulich will also speak this afternoon. So the vision for the vault, for the collection, is really to have a non-profit, non-government foundation where the collections can have a backup repository, and also build capacity across the world. And to do this on the collection side, the idea is that depositors will, just like a bank, will deposit their collection, having the exclusive rights to move it. Every deposit will be sequenced, and the sequences made available for any scientist to work with it. So it's an open access. And then the collection will be preserved in a safe, secure place. On the other hand, part of the capacity building is the GloMiNe Symposium, and we want to expand workshops also to be more specific, for example, data analysis, etc., to really expand the capacity to analyze data globally. The workshops will always center on the microbiomes, human and environmental, anthropology, ecology, ethics, and techniques. So the website, we have a new website that you can see. It has more information. And I thank you very much for your attention, and I'm looking forward to answer your questions.
Thank you so much for your nice presentation. So I would like to give the chance for the participants to ask a question. So you can use the Q&A.
Okay, I don't see any questions yet.
So I don't see. So Alex, I have a question.
Yeah, Gloria, thank you very much for the nice presentation. My first question will be, you talked about one of the aims of the Microbiota Microvault will be to help build capacity, and particularly once you are dealing with this symposium in Africa for the first time, where we have very talented young people. What are some of the plans that the Microbiota Vault, what are some of the plans that you have in mind when you talk of capacity training, what goes into it so that the young people can explore, so we can build a strong team in the future, people who can be a cadre, supporting and helping to preserve the microbiota in that region. Thank you.
Yes, thank you. It's a very good point, and we believe that only educating and leveraging the capacity globally will local collections be possible. So local collections our effort is different to the effort of the Microbiota Conservancy, in the sense that the basis of everything is local collections. And because the wealth of the diversity is in developing countries, that's where we have to put efforts to capacity building so that those local collections are viable. And those are the working collections where scientific research will be done, because the centralized one is just a backup. So we have planned the GloMines. This is the second one. We plan to do one every year in different parts of the world. So you are welcome to propose places. The idea is to create and stimulate and foster the efforts for local collections in the places where the GloMine happen. Indonesia is in line, and Guatemala are also queuing. They have interest in doing GloMines together with them. Another thing we have thought is capacity to analyze data, because I recognize, and I understand very well, being South American, that sometimes it's very hard to get money to do research because the reagents are expensive, because you don't have the latest technology. Sequencing is changing so fast and is so expensive. But the world is getting more and more and growing in data. And you can mine that data and do research. So the capacity to analyze data, which requires a good computer and a good internet connection, that is cheaper in real terms than running a lab. And so we think that if we leverage the high-tech capacity of the richer countries with the human capacity to not only collect, but also analyze data, then we can make an ideal synergic relation that is very productive and beneficial for both sides. So I do believe that capacitating in data analysis and protocols, standardizing protocols is extremely important. And that's one effort we will try to make, more workshops on sequence analysis, etc.
Thank you very much.
Thank you so much. I think you have a few questions on so I'm going to ask you one question. They say is, Kobla said, in restoring the health microbiota to individuals who have lost due to the antibiotic use and westernization. So what are the plans of the Vault in contributing to this?
So again, the Vault will support local collections where needed and keep a backup, like in a bank. Only the depositor can move that. Now, suppose we learn how to restore, because right now, apart from the fecal transplant to cure infections by Clostridium, we really don't know how to restore, and especially how to restore early so that we can prevent. But the day we know, then we will know where are these microbes. And again, the backup, we hope, will not be touched for many years, but the local collection that presumably has stored that diversity has it. So the idea is that research will be done with them, and the countries and the local scientists will have probably propositions from industry that they will have to deal with their governments, etc., to develop new medicines to prevent, or new probiotics, etc. So we wouldn't deal with that. It's a completely, entirely the capacity and say of the local collection. There is another question, from Laila, what is the origin of samples included in the vault? Everybody site and environment, yes. So we are starting in the pilot because we have to start small with the gut and food, fermented foods from the world. We hope to expand to the environment, plants and the environment as well. Great, Teresa Noguera says, great project, would like to know if the metagenomic data of these metagenomes will be freely available. Yes. So the idea is every deposit, and that would be a condition, pretty much, we think, that people should agree that to have a free metagenomic sequencing of their sample, of their specimen they want to deposit. So they are offered a backup, but also metagenomic sequencing with the condition that it can be open access so that any scientist in the world can do studies. So if you imagine in the long run, there will be metagenomes of complex samples from many body sites in different regions or environments in different parts of the world. And many studies can be done together with the metadata, of course. So that will be an effort to a good database with the metadata.
Thank you. So we still have more 5 minutes, so I will allow the participants, if they have more questions for all the panelists, so they can ask. So Karina.
Oh, Selena has another question. Do I have time to answer it?
Go ahead. We have
Sure.
time.
Yeah, you mentioned that for animals, what are the specifics if one is also interested to work on animals. And Karina, are there studies that you think on the phylogeny between the organisms originated from different places? So yes, we are interested in basically any microbes that are important to humans. So microbes from animals are important to humans because either they are agriculturally important or because they are important for the environment and the ecology of the planet. So in general, that means every microbe will be important. So if there are animals of conservation interest because they are in danger, or because they are wild, or because they are we use them for animal production, any microbes that are valued by the depositor, the depositor can request to make that deposit of the samples. We will standardize protocols, recommend standard protocols across, and we hope to have that on the webpage when the time comes. Part of the pilot study is also studying the different methods of preservation, and eventually we will have the protocols available online in the webpage. Yeah, I think that those are, and yes, anybody will be able to access the data, ask questions, do phylogeny of the microorganisms, study one microbe if we have the whole genome sequence, and see how is it distributed, in the world, and etc.
Karina, we have more 3 minutes. You can ask.
Gloria, when we started to prepare the symposium and we were talking about the microbiota vault, some people expressed the concerns of the ethical regulations that they had in their local countries, and that are different from country to country. And in some cases, it's very difficult to get permission to store samples, microbial samples and diversity samples. And maybe it would be important to have some kind of access to some consultation to help in these cases? Do you have any idea how the vault could provide some guidance for ethical agreements?
So, every country, as you say, is different, have different regulations. The idea is to form a consortium. It's not to centralize in the vault, but right now we are interconnected. And the idea is that if there is a similar system, if an African country is similar to Peru, for example, we already have the network and we can help connect or help also, find out with lawyers if international law is required, etc. I think the most important is really to have the local collections. Once you have the local collection, the first step is done. And then we have to study how do we navigate from that particular country, that particular system with their ethical committee and etc. regulations, how to make the backup deposit to the vault. But at least it's already preserved somewhere. The urgency, what is really, really urgent is to start the local collection. And the Microbiota Vault is willing to help support the local collections where needed. Liquid nitrogen, which is much safer than electric, ultra low temperature freezers, especially in developing countries where the electricity is so unreliable everywhere. And, you saw what happened to my collection in New York. It's not, if it was a liquid nitrogen, then we will have to have a provision of refilling the liquid nitrogen. If there is a need for tanks or some support, we will help finding that support for the local collection. Then from there, each country will have a different story, how to deposit in the central vault, and we will navigate together.
Thank you so much. And I would like to thank all the speakers